Let us continue with the formation of blood with the explanation or exposition. So, cells that are responsible to do function of hemopoiesis are first seen in the yolk sac of embryo in the third week of embryonic development, and these cells are known as hematopoietic stem cells. So what are stem cells? Stem cells are special human cells that are able to develop into many different cell types. Hematopoietic system. So it consists of all organs and tissues that is involved in hematopoiesis, and these are divided into the myeloid tissue and the lymphoid tissue. The pluripotent hematopoietic stem cells, or HSC, is the progenitor of all the cells in the blood and gives rise to cells of the both myeloid and the lymphoid system. The stem cells, after a series of division, differentiate into two types of progenitors. We have the lymphoid stem cells and the non-lymphoid or myeloid stem cells. We call it as the trilineage, and for lymphoid we call it the immune system stem cells. So for megakaryocytes, we have the colony-forming unit megakaryocytes, or CFUMG, burst forming unit of the erythroid line as BLUE, and colony forming unit granulocytes and macrophages as CFUG. And lymphoid stem cells further develop into T, B, and NK cells. NK means natural killer cells. So the non-lymphoid or the myeloid stem cells differentiate into three types of cell lines. We have the granulocyte monocyte progenitor that produces neutrophils, eosinophils, basophils, and the monocytes. We have the erythroid progenitor that produces the red cells, and we have the megakaryocytes as a source of platelets. Here is a picture that shows normal hemopoiesis. So we have the pluripotent stem cell, from where it is further divided into two types, two lineage. We have the myeloid stem cell and the lymphoid stem cell. From the myeloid, it is further divided into three portions. We have the granulocyte monocyte progenitor, erythroid progenitor, and the megakaryocyte progenitor. From the granulocyte monocyte progenitor, we have the neutrophils, basophils, eosinophils, and the monocytes. And from the erythroid, we have the red cells, megakaryocytes, we have the platelets. So this will undergo a series of transformation. So I'm not putting it here. So let's look into lymphoid stem cells. We have three types from the lymphoid series. We have the B lymphocytes, T lymphocytes, and the natural killer cells. B lymphocyte formation takes place in the bone marrow, T lymphocytes in the thymus, and NK in the bone marrow, natural killer cells in the bone marrow. So this is a series or the hierarchy of blood formation. We have the stem cells, from where it gets differentiated into multipotent progenitor cells, and further into committed or unipotent progenitor cells, and further into precursor cells, and later on the mature cells. And for the differentiation and maturation is regulated by endogenous glycoproteins, cholesterol factors, cytokines, and hormones. For example, we have the erythropoietin that regulates erythropoiesis, granulocyte colony stimulating factor for the granulocyte, and the granulocyte macrophage colony stimulating factor for the granulocyte macrophage lineage, and thrombopoietin for formation of hemopoiesis. So those are examples of the regulatory systems of hematopoiesis. I hope you have understood. Let's look further into the summary of this topic.